Good morning or afternoon or evening, whenever you're watching this. But here we go with cellular respiration part 3. It's like I know you've just been hanging on to your seat waiting for the end of the story. Well, here it is. We're starting today. All right, maybe. Okay, here we go. Couldn't get the slide to change. All right, so remember the big question here was, is there oxygen present or is there not? Because there's two very different paths we could take. In both aerobic and anaerobic respiration, we're both going to have glycolysis. So we have glucose broken down into pyruvate, and that's occurring in the cytoplasm. But if we had, what we looked at before was if we had oxygen, we went into the mitochondria and we went into the Krebs cycle and then the electron transport chain. So we had ATPs produced in glycolysis, ATPs produced as a result of Krebs, and ATP produced as a result of the electron transport chain in the mitochondrial membrane. But now we're going to look at the other side of the story. If there's no oxygen present. If there's no oxygen present, we're either going to make ethanol or lactate. So let's look at the alcohol. All right. So in the absence of oxygen, we're going to undergo fermentation. And like I said, we can either have alcoholic fermentation or lactic acid fermentation. And with fermentation, this is going to occur in order to free up the electron carriers in order to keep glycolysis going, because the ATPs are only going to be produced here in glycolysis because there's not going to be a Krebs cycle or an electron transport chain. And so we're going to have, oh my goodness, I just looked outside and I saw my ferns are growing like crazy. Whoa. Sorry, little digression there. All right, so the ATPs are only produced from glycolysis. And so there has to be a means of keeping that going. So these the electrons that are stored here in NADH are released, and we're ultimately able to make ethanol. And when those bonds are broken, then that energy is used to make ethanol. But then that electron carrier, the NAD plus, is able to go back through this cycle and keep glycolysis going and keep ATPs being made. All right, so in alcoholic fermentation, it's going to occur. We're going to have this in bacteria, we're going to have this in yeast, and we are converting, of course, we're still going through glucose and then we're going through glycolysis, but we're converting these two pyruvates ultimately into ethanol. So you want to keep in mind, oh, I'm so sorry that I just sneezed into the recording. All right. All right. So we're going from glucose to pyruvate to ethanol. You don't have to memorize the any of the intermediates here, the structures or anything. But I do want to point out that the CO2, the carbon dioxide, is given off here because we're still staying with our formula of the products being water and carbon dioxide of cellular respiration. And here's where the carbon dioxide is released. And so CO2 is released, and then we produce the ethanol. Beer, wine, bread, these are all types of alcoholic fermentation. They all undergo this process. So glucose, pyruvate, CO2 given off, and ultimately we end up with ethanol. Now the other type of fermentation is lactic acid fermentation. Same kind of process. We're going glucose, pyruvate, but now we're going to lactate. And you can see we have in lactic acid fermentation, again, ATPs are being produced from the process of glycolysis, but we're not having any of the Krebs or electron transport. And so now pyruvates are being converted to lactate instead. It's breaking a double bond with the oxygen here and adding a hydrogen on. So we've gone from a carbonyl group here to we have an alcohol group here and a hydroxyl group. And cheese, yogurt, muscle cramps. These are all types of fermentation, of lactic acid fermentation. Now, the other thing that you need to know is there's a process, like this doesn't continuously happen. If there's enough ATP, then there's processes in place. It's called a negative feedback loop, and it will stop. It's like an on-off switch. And so this phosphofructokinase, kinase meaning ACE, meaning this is the enzyme, and so this phosphofructokinase is the on-off switch. So you can see here we still have the process of, and of course this diagram is showing aerobic respiration because we've got glucose broken down into pyruvate and then going into the Krebs cycle, and then the electron transport chain, and ATP being made from glycolysis, Krebs, and electron transport. So if there's plenty of ATP, then that's going to signal phosphofructokinase to shut down. It's not going to be active. Or if there's enough citrate coming out of the Krebs cycle, that's one of the products that, one of the conversions when we looked at that all those different molecules that are made, intermediate molecules. And so if there's enough citrate, then also that's going to inhibit the phosphofructokinase. And what stimulates it is adenosine monophosphate. And so if there's adenosine monophosphate, it's going to go ahead and start that process because it's used up the other phosphate groups. So here we would have adenosine monophosphate. If we put on the second ATP, then this will shut this off or turn it on. If the second ATP is not used, then the glucose is not going to break down into the G3Ps, the glyceraldehyde 3-phosphates. All right, and it's not going to go through this process. So that's our inhibitor. And then the other thing, oh, it's not on here. Whoa, we lost it all. That's way back there. Oh my goodness. Okay, so the last thing to talk about is the facultative anaerobes. And the facultative anaerobes, these are both aerobic and they can perform both aerobic and anaerobic. So they can go through either one. So they're really versatile. They prefer oxygen because it produces more ATP. It's getting ATP from glycolysis, Krebs, and the electron transport chain versus the ATP being just formed from glycolysis and anaerobic. But facultative anaerobes can go either way. That's all I have for you today. End of the story. How exciting. Have a great day.